Hello, we're going to go through Competency 3 on cellular metabolism. The WID's objectives for Competency 3 look like this. We've got protein synthesis, which we're going to discuss in Competency 5 as well. So I'm going to just introduce you to a couple of key features in this particular presentation, because we do go through it in a little more detail in Competency 5 as well. So it must be important. Aerobic and anaerobic respiration, metabolism, including anabolism and catabolism, carbohydrate, lipid, and protein metabolism, cellular organelles and metabolism, and cellular membrane transport. So we're going to hit all of those bases as we go through this particular presentation. This is the third competency. So we've gone through nervous system and some of the basic physiology. So we've looked at a little bit of cellular and ionic features, including membrane transport. We looked a little bit at sodium moving into a cell, a neuron cell or a nerve cell, to elicit some sort of change in electrical activity and potentially leading us into some sort of action. So we've seen sodium moving into a neuron, potassium moving out. So we've looked a little bit at cell transport mechanisms. In general A&P, you looked a little bit at some of the cell organelles and maybe touched on a few of the basic functions of those organelles. So we'll go back and look at that again in this particular presentation. So the foundations of physiology and a lot of what you're going to be doing in the healthcare field relate to pharmacology and analyzing through series of different diagnostics what is going on in the body. So at this level, the chemical level and the cellular level is really where we're going to live throughout this semester, and really kind of where you're going to be in the clinical setting as well. So this is some foundational information, and again, some of it you've seen in general. So I'll keep it relatively brief for that reason. As a review, we look at elements and compounds. Again, we're at the chemical level. And remember our levels of organization. The most basic general A&P where you started we're at the chemical level where we're dealing with different elements, different ions, different molecules, different compounds, bonding, all of that leading us to the cellular level of organization, and then the tissue level, the organ level, organ system, and finally the organism level. So again, in general, we stuck with primarily the organ systems and the organs. Very little with tissues and cells, and even less with chemistry. So that's what advanced is for, is the chemistry and cellular portion. So when we look at the chemistry and the elements that are primarily involved, we look at carbon, hydrogen, oxygen, and nitrogen. C, H, O, and N are the four primary elements. They make up roughly 96% of the total elements in the body. So there are 22 others in addition to these four that make up the other 4%. So 4 of them make up 96%. 22 of them make up only 4%. So really, when we're dealing with biochemistry throughout, we're going to see these 4 elements, C, H, O, and N. Okay. So we'll talk about that in more detail, and you'll see those as a consistent theme. Here's the periodic table of elements that also shows with some highlighting what those 26 elements are. So we have what are called the major elements and the trace elements. So in addition to H, C, and O, we also have a couple other major elements, sodium and potassium. We've saw these when we were going through the nervous system. Magnesium and calcium, two big ones that hopefully you're consuming every day in whatever foods and maybe even supplements that you're taking. And then we see phosphorus, sulfur, and chlorine. So the Cl, chlorine, we oftentimes see connected with and kind of following along with sodium. So that NaCl combo, a table salt, also in the body shows us that sodium generally is found in large amounts in the extracellular fluid or outside the cell. That also happens to be where we find chlorine. And the chloride ions will follow sodium ions generally wherever those sodium ions go. Potassium, again, we generally find living inside of the cell in that cytoplasm. But again, these are not all or nothing. There's always a little sodium in, there's always a little potassium out. So at least hopefully. And then we have some of the other trace elements, boron, fluorine, aluminum, silicone, and a few others. Our main focus really throughout this semester is going to be on carbohydrates, proteins, and fats, which are going to include the CHO as well as N's for proteins. And then we'll also be discussing throughout the semester, we've already started with sodium sodium ions, chlorine ions or chloride ions, and potassium. We will also see some magnesium, calcium discussion throughout the semester, and we'll get into a little bit certainly with sulfur and phosphorus. I'm sure all of you have heard those three magic letters for energy, AT and P. So the P of ATP is going to relate to phosphates. So that will come up a little bit later in this discussion. So again, in the cytoplasm, we're going to see CH and O. We also see iron and manganese and silicone. These are just as side notes. These are in the cytoplasm. Again, a lot of these trace elements as well as some of these majors are going to be found more in the extracellular, magnesium, calcium. It just depends on whether we're getting a stimulus and an action or not, or if the cell and the tissues are at rest. So again, we've already seen some of this going. So I want to go into briefly what a chemical reaction is. It's either the formation or building of something or the breaking apart of something. So really when we talk about chemical reactions, we're also talking about the word metabolism. So keep that in mind. Whenever you hear the word metabolism, it simply means a chemical reaction. Now, there are two major types of chemical reactions. There are four total, but I got rid of the other two so we could just focus on the main two, synthesis reaction and decomposition reaction. When we synthesize something, we're building it. So we also know this as anabolic or an anabolism situation. A decomposition reaction is when we're breaking something down, so we're breaking something apart. So we oftentimes refer to that as a catabolic reaction or catabolism or catabolism. So we'll look at both of these in a little bit more detail. So catabolism, the actual breaking down of a large molecule into multiple small molecules occurs usually in the presence of water. So when we have a compound or a large molecule that needs to be broken into smaller molecules, one of the main ingredients to get that to happen is we have to add water. So we see this happening when we digest food. So in a few competencies from now, I believe it will be in the last module 4, we're going to do digestion and nutrition. So we'll look at some of the other features that allow our bodies to take a piece of steak or take a potato or an apple and break those large chunks into smaller usable chunks. So that's really the goal or the aim of consumption, is to be able to catabolize or break down large macromolecules into smaller usable molecules. So that's called catabolism. And again, one of the first things we need to add to get that chain of started is we need to add water. So again, it's important to always consume water. Our bodies generally are relatively hydrated as it is, unless there are other external factors of consumption going on, or some sort of disorder with the kidneys or the liver that may be altering our hydration. So anyway, water. Add water to something, it's going to start the process of breaking it down. What water does, it's called hydrolysis. We see the end of that word, lysis. Lysis just is a process of breaking something down. Hydro means water. So we add water, we break something down. That's what hydrolysis means. The other thing we need to add, and we'll see this later today in this presentation, is the addition of an enzyme. Enzymes also speed up reactions of breaking down or catabolizing. So whenever we need to go through catabolism, water is important, as well as enzymes. We're going to go from a big chunk of protein or a steak and ultimately get to amino acids. We also see catabolism going on at the cellular level. So inside of an actual cell, we also see the breakdown and chemical process of catabolism. Okay, so we'll see, it will look a little bit at cellular respiration and the gaining of ATP from glucose or other carbohydrates, primarily glucose and oxygen as part of that cellular respiration. The breaking of this phosphate bond, we have T meaning tri and P meaning phosphate. So we have 3 phosphates. So when we break that bond, we add water to ATP, we break that bond. That bond breaking is what provides energy at the cellular level. So anyway, catabolism, breaking something down into smaller bits, or breaking a bond to form energy. So anyway, breaking something apart is catabolism. And again, sometimes when you break something apart, you create or you gain, not really create, but it releases energy. So when I break a bond, energy gets released. So that's a type of catabolism, and that goes on with the mitochondria. We'll look at that a little bit later. So again, we can either take a big chunk and break it into smaller chunks. Catabolism. It usually involves adding water. We call this hydrolysis. If we dehydrate so we have a lot of small molecules contained or floating around in a watery environment, and we remove or dehydrate, pull some of that water out, what it can do is open up some binding sites for these small molecules to actually attach and form bigger molecules. So we oftentimes see this with something we call protein synthesis. So an anabolic reaction is a building of some of a molecule from small bits, putting those together and getting a large chunk. So it's the opposite. These are reversible. So catabolism goes from big to a lot of smalls. Anabolism goes from a lot of smalls to a big. So that's those are the key features that you really want to remember with these. So whenever we hear the word synthesis, we're thinking of an anabolic or a building up of something. And again, that's going to involve the opposite of adding water. It's going to involve taking water away or dehydrating to allow small molecules to become large. So when we look at this diagram or illustration of metabolism, if we start over here on this side, we have a polymer, which is multiple, we have multiple bonds. We add water. So this is a large molecule. We add water to this large molecule, and it's going to cause a bond to be broken. So we call that hydrolysis. Add water, lyse or break a bond. So this big polymer becomes two smaller items. So we see one of the hydrogens take over this spot here. And then we had this chunk left over that we broke apart. So the OH, the other hydrogen, so we have H2O, so we have two hydrogens. One attaches to this chunk, the OH, the other H, and then the O attached to this chunk. Okay, so that's hydrolysis. We took a big chunk, added water to it, and got two smaller chunks. Now when we move over here, we take these same two chunks. So that's what this arrow is saying. We're taking these two items. We're going to move them over here. So that's what we have going on. This time we're going to take the hydrogen and the OH group over here. We're going to break those bonds apart, and we're going to dehydrate. So we're going to pull water out of there. What that does is it frees up this spot. So this molecule can join up over here, and then we get our polymer or our large molecule. So we've just got this cycle that's continually going on where we're consuming something large, adding water and enzymes to it, getting smaller bits. Then the cells are going to take these smaller bits. So if this is the protein steak, we break that bond and form multiple amino acids. Then in the cells, we can go through protein synthesis and dehydrate these bonds and allow for the production of some sort of protein, usually in the form of maybe an enzyme, a hormone. We looked at hormones. A multitude of hormones are protein-based. Remember, we had our steroidal and non-steroidal group, and that steroidal group was lipid-based. We only had about 5 or 6 of those, and everybody else was on the protein side for hormones. So we can create hormones in this way. We can create enzymes in this way. We can create protein structures in this way. So basically anything in the body that is a structure is going to be made of protein. For the most part, we do have some lipid-based or some carbohydrate-based, but there's usually a link to protein in there somewhere. So the idea is we take a large protein, add water and enzymes, we get smaller amino acids, then we can add those amino acids together and create a new protein. So you are what you eat is basically what this is saying. You eat protein, body breaks it down, you build proteins. That's the basic foundation of life and of metabolism. Okay? So again, chemical reaction is the fancy, or metabolism is the fancy term for chemical reaction. And we see two major types. Okay? Decomposition or catabolism, okay? Synthesis or anabolism, breaking down the large to get smallers, building up the smallers to get a larger. Okay. Again, just a couple quick black and white of it all. We're going to hit a couple of the objectives just within this, cellular respiration, we've looked at a few minutes ago as an example of catabolism. When we look at cellular respiration, we're primarily talking about glucose and taking that macromolecule. So going back to our objectives, we're going to look at glucose, lipid, and protein synthesis, glucose or protein metabolism. So glucose metabolism typically comes in the form of catabolism, and this pathway for glucose metabolism is going to occur inside of the cell. So the idea is we consume some sort of carbohydrate. We break that carbohydrate, so we catabolize. We break the carbohydrate into smaller bits. Glucose is one of the most usable small bits of carbohydrate that we are going to see. So we deliver that glucose within the bloodstream, drop it off at the cells in the body. At that point, the glucose enters a cell. It's in the cytoplasm or inside the fluid of the cell. This is where we see what's called anaerobic respiration. So glycolysis is the first pathway of cellular respiration. And again, that occurs in the cytoplasm. Anaerobic, no oxygen needed. We're going to get a couple of ATPs from this reaction. At that point, we're going to move everything, what they call pyruvic acids or pyruvates from glycolysis. They're energetic. They're going to go into the mitochondrion, okay, or mitochondria is the plural. So in the mitochondria, we're going to add oxygen to the mix. At that point, the oxygen and the pyruvic acids that came from the original glucose are going to continue the process of getting us to making optimally about 36 ATP molecules. So when a glucose enters into a cell, it's going to start being metabolized in the cytoplasm. Okay? We don't get very much ATP from that. Without oxygen or in an anaerobic situation, we're only going to get a couple of ATP. Okay. And during this glycolysis process that doesn't use oxygen and only gives out a little bit of ATP, it also gives out a byproduct called lactic acid. So if we stay in glycolysis for too terribly long, mainly because we don't have enough oxygen, we're in what we call an oxygen debt. So if we have an oxygen debt, then so that happens during working out or doing activities that maybe you're not used to doing, and so you haven't built up an oxygen. You haven't added your oxygen. You haven't built up your oxygen bank enough. So that's why if you haven't been exercising or moving around or doing a lot of physical activity, and then all of a sudden you do. You tire out fairly quickly. You may have muscle soreness the next day or even that same day. It's just because the requirements that you have for your muscles to perform activity outweigh what the body's ability to provide oxygen and glucose to the mitochondria. It won't happen. So your needs outweigh what is on the supply that you have. So the demand is outweighing the supply. At that point, the anaerobic respiration will occur and continue to occur because we do get some ATP, but we also get lactic acid, which cause muscle soreness. So anyhow, if we get all the way through the three pathways, we should be able to get 36 ATP if we involve oxygen. And again, that occurs in the mitochondria. Anabolism, we think of protein synthesis as a central anabolic pathway. We're going to go through that quite a bit in Competency 5 because Competency 5 focuses on DNA and RNA. So DNA and RNA um, are the building blocks of the blueprint of the cells and what the cells are made of and what the cells are supposed to be doing. Basically, the nucleus contains all of the key information in the form of DNA to guide the cell to go through protein synthesis and lipid synthesis. So when proteins or amino acids enter a cell, we're going to see this process of transcription and translation of the DNA being photocopied in the nucleus, delivered out into the cytoplasm of the cell, where we're going to find ribosomes and rough ER working together to basically read and translate what the nucleus wants to be produced. So proteins, amino acids are going to line up and the ribosomes and some of the messenger RNA, the transfer RNA, and the rough ER, all these R's, the rough ER, the ribosomes, the RNA, they're all going to work together to read the blueprint that came from the nucleus and produce some type of protein. So that protein could be in the form of an enzyme, a hormone, or a structure. All right. So the primary organelles, ribosomes, and rough ER for protein synthesis. The smooth ER will help with lipid synthesis. So carbohydrates go to the mitochondria, proteins go to the ribosomes and rough ER, and lipids go to the smooth ER. Just a couple FYIs. Again, we're dealing with carbs, proteins, and fats. Carbs and proteins are going to have carbon-carbon bonding or carbon-hydrogen bonding. That's what makes them organic. So something that's organic molecularly is going to have a carbon and a carbon bond, or a carbon and a hydrogen bond. If there is no carbon-carbon bond or carbon-hydrogen bond, it's considered inorganic. So NaCl is table salt. So is NaCl inorganic or organic? Well, we have Na, which is sodium, Cl, which is chlorine, coming together. Well, I don't see a carbon or a hydrogen in any of that. So that NaCl would be an inorganic compound. Whereas glucose is going to be C6H12O6 in its formula, C6H12O6. So we have C's and H's involved. So that's going to be an organic molecule. A couple other inorganics, oxygen, which is O2. We don't see any carbon or hydrogen there. Carbon dioxide, that's a tricky one maybe. It does have carbon, but it's CO2. So there's only one carbon and there's no hydrogen or other carbons. So we have one carbon and two oxygens. So again, that doesn't fit with the carbon-carbon or carbon-hydrogen group. A lot of our electrolytes are going to be inorganic, your magnesium ions and calciums. Okay, so the organic group, we're going to see carbs, lipids, and proteins. Carbs are your sugars and starches, and we primarily find that term saccharide when we look at carbohydrates. So the idea is we want to allow our mitochondria to have some monosaccharides. Glucose, fructose, and galactose are the main monosaccharides. And if we put a couple of glucoses, or a glucose and a fructose, or a glucose and a galactose, or a fructose and a galactose, if we make any combination of two of these monosaccharides, it forms a disaccharide. Then if we string more than two together, we have what's called a polysaccharide. So we'll look at those in more detail in a little bit, but we do see carbohydrates very simple, um, primarily used for energy purposes. Lipids, a little more complicated. We can see kind of a triple group coming off here. This is what they call a triglyceride. So glycer we see with fats, saccharide we see with carbs. And then we get to proteins, and we're going to see that these are a lot more complex, and so a lot more going on with a protein. And again, the glucose is the primary unit of carb that we're going to use for energy. So we can see we've got a six-sided figure here, 6 carbons, 1, 2, 3, 4, 5, 6. So we have a 6-carbon sugar or glucose. And again, here's the formation of. So formation, this would be synthesizing, which again is going to be a dehydration reaction. So we take two, remember synthesis, we're taking smaller bits and making a larger bit. So we have 2 small bits, a glucose and a fructose coming together. And we remove the H and the HO, or what they call the OH group, the hydroxyl group, and then the hydrogen. We combine those HO and the H, we get H2O. So we dehydrate. What that does is opens up a bond here. We can form glucose plus fructose equals sucrose. And again, we can go the opposite way and do what's called hydrolysis. So we can add water to this, and it'll break that bond, as well as some enzymes. Sucrase enzyme and water will break that bond and create glucose and fructose. So this is a reversible reaction. Sucrose, by the way, is white granulated sugar that would maybe come from sugar cane or sugar beets or something like that. And we'll talk more about carbs, proteins, carbs, fats, and proteins at the end too, when we get to digestion at the end of the semester. Lipids are fat-based, and again, these are going to be energy sources. This is stored energy. We also find structural aspects of lipids. We would see our cell membranes, okay? And then hormones, right? Our steroidal or lipid-based hormones. And again, we see this glycerin here, so triglycerides. Okay. And fatty acids come from the breakdown of a triglyceride or a fat. And these triglycerides or fats are going to be saturated or unsaturated. Saturated means we have a solid at room temperature. And notice we have no carbon-carbon double bonds anywhere. So that's what makes this polarized and allows these little glycerols to come off of here. And then we have a couple carbon double bonds in here, so that you can see that going on here and right here. This is what they call an unsaturated fat. Okay? Those would be liquid oils at room temperature. And again, we see the formation of, so this would be a synthesis or a dehydration reaction. So we're getting rid of water. We have 3 fatty acids. This OH oftentimes is what's coming off with the H of another to form again. We have 3 of them, so we're going to add 3 water molecules. So we're going to have a glycerol attaching to 3 fatty acids, forming a triglyceride. And again, this can go the opposite way, right? We can do a catabolic or a hydrolysis reaction where we can add water to the mix and cause these, as well as enzymes, of course. Lipase are special enzymes that help to break down triglycerides as well. And we see, just as a reminder, in the cell membrane, we call that a phospholipid bilayer. And so we see that one end of the phospholipid is going to be water soluble, so what we call hydrophilic. And then the other end is going to be fat soluble or hydrophobic. Generally, the hydrophilic ends are going to be the heads that are going to be facing toward the outside of the cell as well as the inside of the cell, because the outside of the cell is going to be extracellular fluid, which is primarily water. The inside of the cell is intracellular fluid or cytoplasm, which is also primarily water. So that cell membrane, those double ends, the ball facing the outside of that phospholipid, the ball facing the inside of the cell of the phospholipid, those have to be hydrophilic. They have to be water soluble. In between the phospholipids, we're going to have the tails kind of coming together like this. That's that inner membrane, that inner part of that double membrane. That's going to be hydrophobic. That's fat soluble. So we see that bilayer. So here's the water soluble portion and then the fat soluble tail. Okay. So here we see that cell membrane, that phospholipid bilayer. We have extracellular fluid, intracellular fluid, hydrophilic heads facing the outside and the inside, hydrophobic or lipid soluble tails facing the inside. So water cannot go from here to here unless there's a pore or a channel, and those pores or channels are going to be made of protein. So water and proteins go well together. So we do have to create a pore or a channel for water to pass in and out. Okay. Other fats we've saw, again, that term steroidal, and then prostaglandins we looked at as well. Remember, those are tissue hormones, in essence, local hormones. So some examples of steroids we've looked at: cholesterol, cortisol, testosterone, estrogen, progesterone. These were some of the main steroidal hormones that we looked at. And then prostaglandins, I mean, I would never put something like this and say, what is it? And you probably would never have to be able to identify, unless you're going to become a pharmacist or something like that. You wouldn't need to know. I probably have told you too much already about organic chemistry. You're probably bored to death. So I'm going to speed it up a little bit. So anyhow, proteins are the most abundant of the organic compounds. So remember, the organic compounds are carbohydrates, lipids, and proteins. Carbohydrates we use for energy, so they're not super abundant because we use them. We do store some of them in the liver and muscles and a few other spots. Lipids, again, are primarily going to be found in cell membranes. They're going to be made into some of specific hormones, cholesterols, and proteins are structural as well as chemical. So we see a lot of structural features: muscle, smooth skeletal cardiac muscle tissue is going to be made of proteins, collagen fibers, bone. Most of the organs you guys looked at in general A&P are primarily made of protein. So in addition to that, we've looked at the hormones that are protein-based. So protein is a big deal. Protein is recyclable too, so we don't have to consume a lot, thankfully, relative to carbohydrates, where we have to consume probably 50 to 60% of your daily intake of calories should be coming from carbohydrates, believe it or not. So we'll get into that later when we get to nutrition and consumption. But proteins are recyclable. Whereas, so those reverse reactions can happen with proteins. We can break protein down to get amino acid. We can build amino acids to get a protein in the body. So if there are important enzymes that we're lacking, but we can potentially get the material because of diet or consumption. Well, we can potentially get those materials by breaking down certain proteins, larger proteins in the form of muscle in the body. So anyhow, amino acids are what proteins are broken down into or catabolized into by the presence of water through hydrolysis, as well as protein enzymes. When we get proteins broken down, in the digestive system, we're forming amino acids. Essential amino acids are amino acids that cannot be recycled in the body. We need to consume those in our diet. Non-essential amino acids are recyclable. So there are roughly 20 different amino acids total. Roughly half are essential and the other half are non-essential. Amino acids always are going to then have an amino group, which means we're going to have a nitrogen involved. There's also going to be what they call a functional group or a radical group. So how you're going to recognize the difference between a carbohydrate, a lipid, and a protein is by the presence of this N with one of the main alpha, with an alpha carbon. So the central carbon or the alpha carbon will have an N with it in a protein, whereas a carbohydrate and a lipid, you're going to have a C attached to the alpha carbon or an H, but not an N. And then this R group or functional group, that's getting into deep organic chemistry. I just threw this in here to show you just how complex organic chemistry is, in case you were wondering. But the R group could be any of these attaching right off of the central carbon. Again, formation of and decomposition of a protein, or the synthesis and decomposition of a protein. Okay? Through hydrolysis, we can form, we add or break down. We add water to this large protein, and we can form several amino acids. So again, big becomes multiple littles. Again, this is going to be a catabolic or decomposition reaction. We also call it a hydrolysis reaction. Add water, we lyse the bonds. Okay. And then we dehydrate or take water away. So now we've moved these serine, valine, tyrosine, and cysteine, which are 4 of the 20 amino acids. So we now have 4 individual amino acids. We can dehydrate or remove the water out of each of these and cause them to then have room to form what we call a peptide bond, and thus forming a large protein. So again, hydrolysis, catabolism, add water, break big into small. Okay, take smalls and dehydrate, pull water away. You're going to synthesize or make something big. You've basically removed hydrogens and oxygen to form an opening for bonding to take place. Briefly, when we look at protein structure, we have 4 levels. So you have the amino acid sequence, and then in the cell, in the cytoplasm with the RNA and the ribosomes, we're going to form a polypeptide and coil that into a helix or a pleated sheet. So it sounds like we're doing some tailoring here. And then those pleated sheets get put together and welded together by attracting forces, forming a kind of a globular shape. Then we add some of those globular shapes together, maximally at 4. Typically, we see then a large globular protein or a quaternary structure. This is going to be like a hemoglobin. Remember, hemoglobin has 4 globular proteins, or 4 of these tertiary structures connected to it. And why is protein shape important? Because primarily, again, the structures of your body, your muscle cells, smooth skeletal cardiac, the collagen fibers that help provide strength, the elastic fibers that help provide flexibility. Inside of a cell, a lot of the organelles are going to be structurally find their structure in protein, or foundations in protein. We know that enzymes and hormones are protein based as well. So we do see protein shape important, especially when we're dealing with enzymes. All enzymes are protein based. So any changes in pH, temperature, other chemistry can alter or denature a protein. So if that happens, like typically the body will take care of that. The body recognizes, we have special immune cells that can recognize when um, proteins have become denatured, and generally stop the process of mitosis or of cellular. Really, it happens during interphase. Um, After that S1 phase and before the G or the growth phase, we can potentially recognize if there's some denaturing going on, and we can pull that cell off the line. If we don't, it can lead to autoimmune diseases or other genetic mutations. So anyway, here's some examples of proteins in the bodies. Antibodies, troponin, which we'll see with muscle contraction. You've heard of collagen fibers, DNA polymerase, and we see that ASE on the end tells us we have an enzyme. And then chymotrypsin, an enzyme as well, that deals with digestion. So denaturing, we have a normal protein, somehow gets exposed to an acidic environment or an extremely basic environment, or exposed to radiation, exposed to extreme temperature changes. That's why prolonged fever is extremely problematic, because we start to see the denaturing of special enzymes and hormones and other proteins, structural proteins, during a prolonged fever, because heat denatures proteins. All right, and then lastly, we get into cell transport mechanisms. Just a couple of quickies here. We've got passive versus active. So you've maybe heard these terms before. I think most of us discuss these with you in general to some degree. Passive transport means no energy is required. We generally see the movement of stuff, right? When we have movement of particles or transportation going on with cells, we either need to move the substance to gain equilibrium on the outside of the cell and on the inside of the cell. We need a balance or equilibrium established. So when we introduce substances or what we call solutes or solids into the mix, we can either move those across a membrane, that's called diffusion, or we can dilute an area, and that's what we call osmosis. Okay, so gaining or finding our equilibrium inside and outside of the cell is going to involve any of these three items and not involve the use of energy. Sometimes when we need to get something out of a cell or bring something into a cell, we're going against a concentration gradient, which simply means with diffusion, we're going to see a lot of substance going naturally, just diffusing to where there are less substances or less like solutes. Or we can just add water and dilute an area. Both are passive. And both are going with their concentration gradient. Concentration gradient simply means going from high to low of substance, or water diluting an area that has a lot of substance. Either way, no energy is required. There, and we're dealing with moving with concentration gradients. Active transport typically, first of all, involves the use of energy, and active transport means we're moving a substance against its concentration gradient. So we're moving substances from low concentration to high concentration. Generally, substances don't want to go against the grain, so we have to involve a pump. So this is where the sodium potassium pump or the calcium pump plays a role. Outside of a neuron, there's always a lot of sodium. Even during some sort of stimulus, sodium's going to rush into the neuron, but there's still more sodium outside of the cell. There's always more sodium outside of the cell. So once the stimulus has left the neuron, we need to get the sodium that went into the cell, we need to get it back out. And so sodium won't naturally go back out of the neuron to where it came from in the extracellular fluid. Because there's still a ton of sodium in the extracellular fluid. So we have to put these sodium potassium pumps on the membrane, or embed them in the membrane of the neuron, so we can take that sodium that's inside of the neuron and pump it against its concentration gradient, pump it back out to where there's just a ton of sodium. But we need the sodium in the cell still. We need it out of there. We want to maintain very little sodium inside of a cell when the cell is at rest. Okay. So anyhow, vesicles, also little tiny vehicles that are going to transport substances from inside of the cell to the cell membrane for exiting, what we call exocytosis. These are called vesicles or vehicles. Those do require ATP as well. Ascites is a kind of like a beer belly that's gone bad. So fluid buildup in the belly to where it's clinically a problem means we have a liver failure problem, more than likely. The liver produces albumin. Albumin is your main plasma protein. And so if the liver has gone below 70% production, you're going to see albumin levels go down. And if we drop albumin levels in the blood, what that means then is outside of the bloodstream, so in that peritoneal cavity or in that abdominal cavity, we're now going to see a lot more substance in the extracellular space relative to this amount of substance inside the bloodstream. In the bloodstream, you have albumin under normal conditions. Outside of the bloodstream in the extracellular space, you're going to have some other proteins, some of it albumin, but some transport proteins. You're going to have a lot of particulate sodium, chloride ions. There's a lot of stuff kind of floating around and living in the extracellular space. And so that osmotic balance that occurs, so we don't either lose fluid from the extracellular space or gain fluid into the extracellular space, blood plasma levels are extremely important. If we drop the amount of albumin in the bloodstream because the liver has failed and is not producing any, what that causes is an equilibrium issue. So inside the bloodstream, we now have less substance. In the extracellular space of the peritoneum, we're going to have more substance. And so water, we can either move substance to gain equilibrium or move water to dilute the area where there's more stuff. So in this case, the substances are too large, the proteins that are in the extracellular space and in the bloodstream, specifically with the peritoneum, the molecules are too large to pass. So what we end up seeing is dumping of water from the bloodstream into that extracellular space, and that causes a large belly and fluid buildup. acetes, they call it. So anyhow, diffusion, a passive process. Lump of sugar, large concentration to small concentration, from high amounts to where there are less. So these molecules want to diffuse and spread to, again, attain equilibrium. So we have a cell membrane where we have 10% stuff here, 20% stuff here. So the idea is, if we have a membrane that is permeable, we can move stuff from high concentration to low concentration till we get an equilibrium, 15 on each side. So the total is 30. If we split 30 in half, we get 15 and 15. So that's what we see is a permeable membrane. We can move stuff. If we can't move stuff because the membrane isn't permeable enough, what we call the molecular weight cutoff. That just means the pore size of the membrane, which you'll see in your PhysioX activity or exercise 1. Any of those activities of diffusion and osmosis and cell transport are going to deal with pore size and molecular weight cutoff. So some of these molecules have a certain molecular weight or certain size that won't allow them to fit through the pore. So albumin, for one, is a very large, has a large molecular weight cutoff. So it's not going to be able to pass in and out or through pores. So what we have to do instead is to dilute an area through the process of osmosis. Okay? So osmosis is moving water when diffusion is limited, we can dilute an area with water. So we'll come back to that in just a second, but I did want to show you in the extracellular space, we've got high solute concentration of oxygen, carbon dioxide, water, urea. And notice urea, not going to get in. Glucose and sucrose, not going to get in. All of these ions, they're not going to get in. Okay, so simple diffusion is going to primarily see the movement of small gases, oxygen, carbon dioxide, nitrogen, and maybe some water through these pores. Okay, so again, low solute concentration. So when I have low oxygen in here, high oxygen out here, it'll naturally diffuse through. So simple diffusion, typically we only are going to see gases, small uncharged particles. These other urea, glucose, sucrose, well, urea is a nitrogenous waste, so we generally don't want that going inside of a cell. We're hoping to remove that, put it into the kidneys and eliminate it from the body. Glucose and sucrose, they're going to need special helpers or facilitation to get into the cell. So they just need an extra helper. And then these ions also, we're going to need some special what we call leak channels for those to pass through. So anyhow, osmosis, once again, we have a 5 over here and a 10 over here. The idea is I want to move the 10 I'm going to move 2.5 of these over here to balance it out or get equilibrium, but I can't because the pore size is too small for these molecules to pass through. So the other option, see, it says impermeant solute. That means that solid can't pass through the membrane. So what we're going to do instead is dilute this area that has more particles. So you're going to see water movement going over to the right side. So we get a 7.5% solution on each side. So the ratio of solutes to solvent is going to be equal, or we're going to attain equilibrium. So that's osmosis. And there's a pressure so because we have the inability to move the particles, but we do have the ability to move fluid across the membrane, there's a pressure that's going to be building up at this membrane. They call it osmotic pressure. That's basically how much pressure exists to dilute an area. Okay. You saw that possibly in general if you did any red blood cell work, you may have seen that normally we have an isotonic situation where iso means same, so we're not going to see any type of excess. Equilibrium is always around, so we see a balance of water in, water out, a balance of substance in, substance out. So if this water, this solution represents the plasma of the blood, what we can sometimes see is an increase in too much stuff in the plasma. So if we have an excess amount of stuff in the plasma, an excess amount of solutes in the plasma, we say it's hypertonic. Okay, so when plasma, or the solution that the blood cell is in, has a lot of stuff relative to what's it. So if we took a cup of salt and put it in in a gallon of water instead of a teaspoon of salt, that would be a hypertonic solution. We'd have way too much salt in the water. And so what's going to happen is the water inside of the cell is going to leave the cell to dilute that area in the extracellular space where we find all of that solute, or all of that stuff. So in a hypertonic solution, we're going to see the cell shrink, or what we call crenate. Okay, in a hypotonic solution, so now we have a solution where there isn't anything in it. There's not enough substance. There's not an equilibrium. So now the inside of the cell has more substance than the outside of the cell. So water from the solution is we call that hypotonic. So not enough stuff in the solution. So what we're going to see is the movement or rushing of water from low concentration of solute to where we see a higher concentration of solute. So water is going to dilute the inside of the cell, causing it to rupture or burst. So again, if we have a pure water situation where we've got no solute anywhere, but we have a lot of solute inside the cell, water is going to go and dilute the area where there's more solute. So, and again, there's a pressure that builds up, called osmotic pressure, when the water is wanting to push against a membrane to travel into it and dilute an area. Okay, the last type of passive transport. So we have simple diffusion, osmosis, and now facilitated diffusion. That just means the pore size or the membrane protein or the channel is too small for the substance to pass into the cell. So we need to add some sort of a channel or some specialized receptor site to be able to allow for these larger substances to pass through. So we saw this with the neuron, with sodium leak channels and potassium leak channels. So sodium leak channels to allow sodium to leak in, potassium leak channels to allow potassium to leak out. Okay. So that's facilitated diffusion. We need to add something. We need an integral membrane protein. Okay. Whereas simple diffusion, certain gases can just pass right through that phospholipid bilayer. Facilitated means we need a helper. Okay, we see what's called carrier-mediated transport. So we may have a carrier. So this is channel-mediated. Carrier-mediated means we need a special carrier. We see this oftentimes with glucose. So we may have a specialized integral membrane protein that has the shape. It's kind of like a lock and key mechanism. We see this with enzyme activity as well. But if we have a glucose molecule coming in, it needs to match up to the shape. And that's where insulin's kind of like the key to allow glucose to attach to this carrier protein. And then you can see then the carrier protein will drop off the glucose into the cytoplasm. So this is outside the cell, and this is inside. Okay, so as we bring in a glucose, as long as it has insulin, it's going to be able to unlock the door and travel through. And again, those are all passive processes. And the main active process, again, we're going to see, first of all, the use of ATP. Secondly, we're going against our concentration gradient. And because this requires energy, we have to balance how often and how much we use it. If we do have problems with sodium and potassium, balance, it could be because we're lacking glucose to be able to activate these pumps. So here's a calcium pump. It works just like a sodium potassium pump. We have a high concentration of calcium here. We've got a low here. How do we get from this to go against our concentration gradient? Well, we need some ATP to pump it. Same with sodium, potassium. Remember, sodium lives out here. So sodium has to go against its concentration gradient. Well, we can get 3 sodiums out and 2 potassiums in. Now, the presence of ATP, it doesn't, basically what happens is, remember, ATP doesn't come in and attach and cause an explosion. What happens is ATP, there's a special enzyme that breaks this ATP bond, one of the phosphate bonds. And when that breaks, you're going to be left with a free phosphate. And that free phosphate, so now we have ADP, diphosphate, and P. So if I add 1 P to 2 Ps, I get 3 Ps. If I break this bond of the 3 Ps, just break 1 P off, at that point, what I have is a phosphate that's able to plug in and basically open or trigger the sodium and potassium pump to operate. Okay, so for every 3 of the sodiums that leave, I can bring 2 potassiums back in. And again, once that phosphate activates the release of sodium, we can bring in the 2 potassiums. And then once the potassiums are in, that phosphate is going to break off. And what it'll do is it may reattach to the free ADP that's around. So we can kind of recycle the ATP. The ADP and the P can be converted back into ATP. Or we have to rely on our mitochondria to have enough glucose to go and oxygen to go through cellular respiration and give us the amount of ATP that we need. Okay, and then lastly, just the active transport process, ATP. That's not what ATP means. ATP means adenosine triphosphate. That's one way to remember that ATP is necessary for active transport processing. All right. So anyway, we have one other need for ATP. Well, we have multiples, but one more for right now, and that's if we want to bring something into the cell, such as a food particle. Cells eat too. They can engulf. They don't oftentimes, if this particle is too large to pass through one of those membrane channels, a cell can engulf that through what we call endocytosis and the use of these vesicles or vehicles. This whole process also requires ATP. And then when we come over here, we run it through some protein synthesis. We have some maybe an enzyme or a hormone or some sort of protein that needs to be delivered out of the cell, like a collagen fiber. We put it in another vesicle. We fill it up with gasoline or ATP, and we deliver it out. They call that exocytosis. So endo is eating, exo is exiting. So both of those require a transport vehicle or a vesicle, and that all vehicles for they require gasoline or ATP. So that's where we're going to see that. So we have a couple different endocytosis. I'm not going to get into too much detail with this. These are just examples of active transport. Phagocytosis and pinocytosis is eating large particles, drinking fluid. Exocytosis, again, is eliminating whether it's waste or even some sort of substance, an enzyme, a hormone, or other protein structure that needs to be released from a cell. So both endocytosis and exocytosis, along with any type of pump, are going to require the use of ATP. Okay, so pumps and vesicle-mediated transport. And lastly, just as a side note, another type of protein that we see is called an enzyme. And enzymes, you want to remember, are always protein-based. And enzymes are chemicals that are going to speed up any of these reactions. So when we go back, so kind of wrapping everything up and putting a pretty bow on top, we started talking about cell metabolism and how do we break something down, a large molecule into multiple smaller molecules. Well, we could just add water to it, but that's not the whole story. We have to add enzymes that are specialized for whatever that large molecule is to allow it to become smaller. So I just wanted to throw in a little bit of enzymes. They don't come up again really until we get to the digestive system, but just know that some of the cellular metabolism does require enzymatic activity, and if we're the chemical catalysts, so if we're lacking enzymes, then that could be detrimental to general cell metabolism. Okay. And again, it's a lock and key mechanism. And the other thing about enzymes is they always end with 3 letters, A, S, E. So if there's an ASE on the end, ACE, ASE on the end, it's an enzyme. Okay, so we have a molecule and an enzyme. So I want to break this molecule into smaller bits. So this is maybe a protein. I want to break it into amino acids, or it's a large lipid that needs to be broken into fatty acids. So we have an enzyme with what we call an active site, and these enzymes are lock and key. So notice this enzyme is kind of an M shape, and this molecule is kind of a perfect fit for that M shape to wrap around it. So that's what we have. So enzymes are specific to certain molecules. So we don't just have one enzyme for all molecules. We have a specific enzyme for a specific molecule. You've heard of lactose intolerance. Lactose intolerance means if I drink lactose, I have a hard time digesting it. Well, you may be lacking the lactase enzyme. So the enzyme always ends in ASE, and lactase is specific to lactose. Lactase may be lacking in some people, or it may be inactive or deactivated for some reason. So anyhow, again, ASE. First part of the word tells what the substrate or type of reaction catalyzed. So is it an oxidase or hydrogenase or dehydrogenase? Hydrolase, so we're going to involve water. Okay. Hydrolyzing enzymes. These are the digestive enzymes. So we're going to see protease, lipase, multiple others as well. So anyway, they're specific in their actions. They regulate metabolic pathways. And again, I just really wanted to throw these in kind of as a side note because all of you have heard of enzymes. Since we're talking about cellular metabolism, in order to speed up a reaction, we must have enzymes. And enzymes are produced in the cytoplasm of a cell through protein synthesis. So one of the items from protein synthesis that we sometimes get is a specific enzyme. Another item we may get is a specific hormone. Another item we may get is a specific structural protein, such as a collagen fiber, an elastic fiber, or something like that. So anyhow, a couple of things, again, allosteric effectors, things that can affect enzyme activity, temperature, pH, those are the big ones. So basically what it's showing is we've got an enzyme here, and then this is the molecule the enzyme attaches to. So this enzyme attaches to something that's kind of a C-shaped, well, a C shape. Well, I may need to make this enzyme inactive under certain conditions. So there's a special uh, uh, what they call an allosteric effector, a protein of some sort that's going to come in and plug in and cause this active site to change. So allosteric causes a change in shape of the receptor site. So that's going to prevent this substrate from coming in and being broken down. So that's an inhibition situation where we want to inhibit an enzyme, right? So there are certain times that we need enzymes to not be working. We want to inhibit the process of the enzyme because we may want this substrate to go through its process and last longer before it starts getting broken down. So sometimes these substrates or these molecules, they have a job that they need to do to carry out to the end before an enzyme comes in and destroys it. So once the substrate has done its job, then we can activate the enzyme. So we bring in a different type of allosteric effector that's going to plug in and rearrange the active site to be functional. So now that substrate or that molecule can go in and get broken down by the enzyme. Just as all this is side note stuff. I mean, you may hear it, you may see it again, but that's it. Again, if you have questions or when you have questions, you can email me, come in for office hours, and we'll get everything figured out and ironed out.